So welcome to Regulation of Gene Expression, part 1. And what I mean by gene regulation is the importance of upregulating or downregulating gene expression to control how much transcript and ultimately how much protein gets made. If a cell tried to keep all of its genes on it all of the time, it would not be very adaptable. And in the case of our body cells, the cell would lose its cellular identity and not work well with other cells in the body. So it's important that cells be able to modulate gene expression in response to internal needs, external stimuli, and signals from other cells. So let's go ahead and transcribe it. NERB cells. The cell uses many strategies to manage gene expression, and we're not going to talk about all of them in this class. We will just focus on 3. First, transcription factors, which we'll talk about today, and on Wednesday and Thursday, we'll talk about histone modifications and DNA methylation. So, yeah. Gene expression, regulation strategies. Again, this is not a complete list. The cell uses many more methods. Your lab this week is actually all about transcription factors, so you'll do some even deeper exploring. Hopefully they're fun activities. I don't know. I have fun writing them. This is often abbreviated TFs, as we've sort of talked about previously. So we'll write TFs a lot. So again, today, talking about transcription factors. So let's talk about what can be found in a transcription factor protein. So these are proteins that can have multiple domains, so multiple functional parts of the protein. So the first is the must-have for anything that can be called a transcription factor must have a DNA binding domain. Now, there are other proteins that help to regulate gene expression, but they only get to be termed TFs if they are able to bind to DNA themselves. DNA binding domain, we always abbreviate DBD, because scientists hate writing the full names for things. And this is a must-have. The other types of domains, usually you have at least one other domain, but it can be a variety of different things, and we'll have a few examples to talk about. So the second type of domain that you may find is a protein-protein interaction domain. In other words, it's what allows the protein transcription factor to contact other proteins. This can come in a couple of different flavors, although there's a lot of ways this can happen. One of the main ones is this domain can help the transcription factor contact the rest of the transcriptional machinery to get transcription initiated. So it might help attract in eukaryotes the general transcription factors. It might help recruit RNA pol itself. Those type of interactions. Transcription factors can activate or repress transcription, as you may imagine, and we'll show an example of each. Another thing that a protein-protein interaction domain might be able to do is to help transcription factors connect with other transcription factors of the same kind. And this is called dimerization, and it's very, very common. So dimerization domains. Oh. The ones we're going to talk about are either form dimers or tetramers, so they like to work in groups. And lastly, many transcription factors have regulatory domains such as ligand binding domains. So the transcription factor may be able to bind DNA or not bind DNA based on if it's able to bind a cofactor or coactivator of some kind. So a ligand is usually any small molecule that can bind a protein. VAP. The two ligands we're going to talk about today are cyclic AMP, a metabolite, and lactose, a sugar. Does it sound like the lac-operon? It does. Get ready. It'll be a new twist on the lac-operon. You won't have to relive your classical genetics class or anything. All right. So first since DNA binding domains are so important to transcription factors, I'm going to tell you about 4 common types. There are more than 4, but these are you will see most frequently in most transcription factors. The first is called a zinc finger. Can be abbreviated ZNF. And this is a small protein domain that coordinates with a zinc ion to help with its DNA binding, and it looks a little bit like a finger and thumb pinching. So that's why it's called zinc finger. Zinc fingers are often modular, meaning one transcription factor may have several zinc finger domains lined up in a row, and it uses that to kind of wrap around the DNA. So I'll draw you a cute cartoon for that. All right, so here I'm going to just draw some little potatoes lined up. Here's the DNA. So here would be some a string of zinc fingers, like 3 in a row. And then they would be attached to some of the other transcription factor domains. We'll draw them as happy potatoes. And I'm going to color the DNA blue. I always think DNA is kind of a blue color, even though it's clear. I like to imagine it's blue. And those are zinc fingers. A second type of domain is also common. It's called a homeodomain. Homeodomains have other names. Homeodomain. Oh, it rhymed. Okay. Another common way to refer to it is a helix-turn helix, based on kind of what the protein structure does. And to make it even more involved, homeodomains are transcribed from genes that contain a homeobox, or Hox genes. So if you ever see Hox genes getting thrown around, this is what it's talking about. It's transcription factors that have this type of domain, DNA binding domain. All right, so in reality, or well, my cartoon version of reality, a homeobox looks like 3 helix that are kind of connected by loops, like this, and then maybe like another one over here. We'll draw it kind of clasping onto DNA. For the cartoon version, I'm just going to draw this guy like a little triangle because it's faster for me. And I will color him in green probably. All right, excellent. So zinc finger, homeodomain, 2 more that I'd like to mention. Next one has the best name. It's a lysine zipper. You may recognize lysine as one of your amino acids. This is what it is, named after the amino acid. And there is a zipper quality to it. Don't worry, you won't be disappointed. Well, maybe. Lysine zippers must bind as a dimer. So you must have 2 different proteins that have a lysine zipper domain in order to grab the DNA. These other 2 you can often find, also bind as dimers, but they don't have to. So here's our DNA. Here is one monomer of the leucine zipper. It kind of looks like an L like that. And here is the other monomer. Here's sort of show it kind of pinching DNA. And then these, the parts that aren't touching the DNA are not hydrogen bonding, but they are zippered together by hydrophobic residues. So this is called the dimerization domain. And essentially, leucine zippers do not like water. So there's a bunch of leucine residues, which is a hydrophobic amino acid. And this is a very sad molecule. And so in order to hide from water, because leucines do not want to be subjected to the cytoplasm, they'll zipper together like this, and then these areas will be able to pinch DNA and bind it. So these guys are happy when they're zippered together. The fourth type, there's four, is the helix loop helix. Not to be confused with helix turn helix, which I'm going to refer to as homeodomain so that they sound different. A helix loop helix also must bind as a dimer, like a leucine zipper. So sort of a similar idea. Here's DNA. One of the dimers can contact DNA with this alpha helix, and then it has another helix here, and they're connected by a loop. The other dimer also will bind DNA, and then will contact, so this is a dimerization. And together they can sort of pinch DNA like a leucine zipper, the same sort of idea. And now I want to color everyone in. I may have missed some. DNA is blue. Boop, boop. Leucine zipper is purple. And I think I made, yes, I think it's like a red color. So. All right, there we have our four types: zinc finger, homeodomain, and the requisite dimers, leucine zipper, and helix loop helix. So just another note just general note about transcription factors and DNA binding domains. So DNA binding domains are not general DNA binders. They must bind at a specific site on DNA that has a sequence they recognize. And so they actually slide around DNA like a creeper looking for their preferred binding site. Furthermore, DNA binding domains will bind double-stranded DNA. So they don't have to open the helix to find their site. They can just peek along the sides of the bases and find where they prefer to bind. Put double-stranded DNA like dsDNA, that's what that means. These specific binding sites are called motifs. So a particular DNA binding domain will have a motif that it would really like to bind to. This can also be called a consensus sequence. They're all fairly interchangeable. And I will give you some examples of specific DNA binding motifs in our next video. And your lab this week focuses a lot on DNA binding sites, and you'll get to see something called a logo, which is a visual representation of a motif. So please enjoy that, and I will see you again in the next video.